Dr. LaRonda was trained originally in chemistry at Rivier College in Nashua, New Hampshire. She went on to PhD training at Johns Hopkins School of Medicine with a longtime friend of the RCSB protein databank, Cynthia Wahlberger, and then to postdoctoral training with another longtime friend of the PDB, Alexander Vlodauer at the National Cancer Institute. Before leaving academe, Nicole rose to the rank of associate professor in the University of Maryland, where she garnered a number of awards and prizes for her work on protein kinases. And today she's going to talk about the unusual RIO kinase ATP family. Nicole, welcome. It's wonderful to have you here in person. I regret in person virtually. I regret that it's not face to face. Thank you very much. Please go ahead.
Thank you very much, Stephen. I really appreciate the opportunity to come here and speak. It's really difficult when you have to think about what to present at the institute that houses the RCSB protein data bank. You're kind of tempted to talk about everything you've put in the database, but definitely decided today to kind of give you an overview of what I've learned over the past few years. Well, not a few, but 15, 16, 17 years of studying the RIO kinase family from a structural perspective. Now, this is work that I did at University of Maryland. As Stephen mentioned, I have moved to Pfizer, but just started about a year ago. So can't really talk about anything that I've been doing there, but I am kind of trying to present a survey of really how structure has guided our studies on the RIO protein family, and hopefully you can get an appreciation for what we learned. So RIO kinases are actually not your typical eukaryotic protein kinases. They, just make sure I get a pointer here. They actually are older than canonical eukaryotic protein kinases, and you can find copies that are homologous to RIO1 and RIO2 from the RIO kinase family in archaea. There's a precursor before the split between bacteria and archaea. And there's actually a molecule that is homologous to both RIO1 and RIO2 that could be more similar to the unknown progenitor of these. These, if you look at eukaryotes, all eukaryotes have a copy of RIO1 and RIO2. And then later on, in multicellular eukaryotes, you can find another protein, RIO3, that is more closely related to RIO1. So really old enzymes that predate the eukaryotic protein kinases. So they're also, although you can clearly pick out that they are protein kinases, they have all the requisite domains, such as the end lobe, the hinge, the C lobe. They are really much more compact and lack all the domains like subdomain 8 that are the activation loop, but also surface for interacting with peptide substrate. So this is a structure from archaea. This is an archaeal RIO1 that was solved a long time ago. Basically now that really showed how unique the structure of these RIO kinases was. So since then, and this is from 2012 now, at University of Maryland, my students, Vatsala Sega and Irene Kiburu, solved structures of eukaryotic RIO kinases. And so here on the left is the RIO kinase 2 from Catamium thermophilum that really looks very similar to what we had solved previously for the archaeal protein. But with each of these structures, we learn new information, and I'll kind of tell you in a minute what we learned from these ones that we didn't know looking at the archaeal structures. But this shows the overall domain organization of the RIO2 protein. There's a winged helix domain, which you may know interacts with primarily nucleic acids, but also that you can have protein-protein interactions. And this helix is unique to RIO kinases. You have this N-terminal lobe. Your alpha C is right here. This is a hinge region that connects the N-terminal lobe to the C-terminal lobe that houses the metal binding loop and the catalytic loop. And in this case, this RIO2 has an extension, a helix on the C-terminus that sort of packs between the N-terminal and C-terminal lobes. We had some ideas of what its role might be at the time. This helix does not exist in the archaeal RIO2 protein. Now in RIO1, if you compare the two, you see the RIO1 does not have this winged helix domain. And this is not the full length. There's a 193 amino acids on the N-terminus of a human RIO1 protein, but it's predicted to be mostly disordered with lots of charged residues. And so no structure has been solved where you can see what's going on at the N-terminus at this point. But one of the key differences is this hinge is very variant compared to all protein kinases. We have this insertion in a hinge region, and what that does is makes for a very deep binding pocket for the ATP moiety, and thus you have really tight binding to ATP for RIO kinases, RIO1 kinases. So one of the first things we figured out when we looked at the RIO2 protein from Catamium thermophilum is that this is a very strange protein kinase in how it transfers phosphate. So in this stick representation here, we have ATP, or ADP, I should say. We basically soaked, we co-crystallized the protein with ATP and magnesium. And in the crystals, what we saw was that the gamma phosphate had been transferred to the metal binding aspartate. And this is unusual, as some of you may know if you're, kinase, familiar with kinases, in that usually the gamma phosphate is transferred directly from the ATP to the substrate. So a serine, a threonine, or a tyrosine from a protein. So that was not the case here. You're seeing this transfer to this metal binding aspartate. We had known from previously looking at the archaeal structures that this residue, which is the canonical catalytic aspartate, seemed too far away to play a role in the transfer of phosphate to the substrate, and realize that there's needed to be some conformational change in order for that to occur. But we, in those structures, in the archaeal structure, we did not see this transfer of phosphate, probably because the archaeal organism we were working with lives at about 80 degrees, and we were solving structures of these proteins from crystals that were grown at room temperature. So we never saw this this transfer. We did some experiments actually to show that this occurs, and that one of the experiments we did was to look at the hydroxyamine sensitivity of this phospho-aspartate, and that would indicate, if it is sensitive, it would indicate that you have this phospho-aspartate in the active site, and we were able to show that we do have this hydroxyamine sensitive phospho-aspartate. And we were really baffled by this because we couldn't understand necessarily why this phosphate remained even in the crystals that had been growing for quite some time. And our only explanation here was that, you know, perhaps this helix that's present in these CT, but not in the archaea, creates this pocket that buries the phosphate and protects it from rapid hydrolysis. So we have a variant mechanism as well, where instead of transferring phosphate directly to a substrate, we have an intermediate step where phosphate is transferred to the metal-binding aspartate. But that wasn't the only thing that was curious. At this point, we had gathered some information that where, you know, there had been a search for substrates. Our group and others had searched for substrates of RIO kinases. Mostly that search was unsuccessful. We also had some information that it could catalyze autophosphorylation, but that reaction rate was pretty slow. So, you know, and then in looking at this, the configuration of the active site and the formation of the phospho-aspartate, we started to think that it kind of was reminiscent of ATPases. So that's when we started looking for ATPase activity and found that RIOK2 and subsequently RIOK1 does have ATPase activity in which it actually releases the free phosphate given enough time. So here's an assay where we're doing basically incubation of radio-labeled ATP with our CT-RIO2, and then looking at the release of the free phosphate, quantifying it. And so this is what's shown here on the bottom. And we looked at the wild type and a mutant of a loop, and I'll kind of point out which loop that is in a second. And then we looked at mutations in the catalytic residue. So D229A is a canonical catalytic aspartate. D257 is the metal-binding aspartate, so where the phospho-aspartate transfer would happen. And you see that these both result in loss of activity, and in most cases without affecting really binding of ATP, which is kind of what this is showing. You might think, well, maybe all kinases might have some background of phosphate release. So we did check that with a few kinases. Here's one where we knew that this kinase was involved in the same pathway. So it was involved in ribosome biogenesis. And we looked at that activity and we did not see release of phosphate for that. So kind of giving us an idea at that time that we do have something that may be more functioning as an ATPase than a kinase. Now it's a very slow turnover rate, about 1 per minute. And that is not surprising considering that we are not looking at the RIO kinase in the context in which it's active, which is on the pre-ribosome. I'll talk about that. So we hypothesized that you would need to have ribosomes present in order to stimulate the ATPase activity. We also learned from these experiments that the release of ADP is slow. So this may be linked to the need to be in the presence of the pre-ribosome in order for you to increase the turnover rate, because the conformation at which the ATP or the ADP is bound in the absence of the pre-ribosome shows a very tightly bound ADP in a deep binding pocket, and it may require opening in order to be able to release the ATP. So RIO kinases are involved in ribosome biogenesis. So I would like to talk about this a little bit, so you have some context. So in brief, you have the production of ribosomal RNA as a single transcript for the 18S, the 5.8S, and the 28S ribosomal RNA, which the 18S goes into small subunit, and 5.8S and 28S go into large subunit of the ribosome. And then you also have this 5S RNA that is separately transcribed by a different polymerase from a different locus. But you have a dedicated polymerase for that. As it's being transcribed, you assemble the ribosomal proteins. You have ribosome assembly factors, things like RNA chaperones, RNA modifying enzymes, and endonucleases that cleave the RNA into the pieces that will end up in a mature ribosome. And this all happens in the nucleolus. The ribosomal proteins, they end up being expressed by Pol2 and then being made in the cytoplasm, and then they need to move back into the nucleolus for assembly. And then you have all of these steps that I mentioned happening in a very coordinated fashion to release the pre-40S and the pre-60S particles. Then these have to be moved into the cytoplasm. You have a nuclear export, and that results in these pre-40S and pre-60S that must undergo further maturation to give you the ATS or the 40S and the 60S that combine to give you the ATS on the mRNA. Okay, so that's a very brief overview. But RIO kinases are required for these last pre-40S maturation steps that occur in the cytoplasm. And RIO2 is required for transport of the pre-40S particle out of the nucleus into the cytoplasm. And this is a very nice review on maturation of eukaryotic pre-40S particles, showing kind of the model for how RIO2 binds to the pre-40S particle in the nucleolus. You get transport. Then it undergoes its catalytic cycle, and that results in the release of the ribosome processing factors that came with the pre-40S from the sort of mid-stage maturation. And then you have further, once RIO2 and these ribosome processing factors are released, you have a premature 40S particle that can interact with 60S particles to give you this early ATS-like particle, perhaps a quality control step. But in that process, you have RIO1 binding in the spot where RIO2 once bound, and that results in further maturation to give you the mature 18S RNA that is the part that is going to be the correct configuration for translation. And in humans, most of this was done in yeast, but in humans, the process seems to be quite similar with very similar steps. The only thing that's not well known is whether or not you actually have this sort of quality control interaction step with the 60S to give you these ATS-like particles. So that has not been seen yet in humans. So going back to this ATPase activity, what we figured on, this was work done in collaboration with Ed Hood and Sebastian Ferrer-Sucker, looking at the role of this ATPase activity on maturation. And what you see is when you make a mutation in the residue, the metal binding aspartate, which becomes a phospho-aspartate, what you see in yeast is that you get an accumulation of pre-40S particles. So you do not end up making, the right ratio of 40S to 60S to 80S. And you see you have a decrease in polysomes, which are, the translating ribosomes. And we saw when you make this mutation, you have cold sensitivity, which basically means that the mutant can survive at warm temperatures, 37 degrees, 30 degrees, you do see some survival, and then at cold temperatures, it's even less. So that kind of gave us the idea that it is some sort of dissociation defect. So looking at this panel here, if you look at the, this is basically a sucrose gradient sedimentation of the ribosomal or the ribosomal particles. And you look for where RIO1 is located in these fractions. You can see that for the most part, in the wild type protein, with the wild type protein, the RIO1 is free, but in the presence of this mutation, you end up with accumulation of RIO1 on the pre-40S particles. So ATPase activity is required for dissociation of RIO1. So when we solve the structure of RIOK2, oh, and I should mention that I showed you data for RIO1, but if you look at RIO2, you see very similar patterns. So when we solved the structure of RIO2 back in 2012, there was also a cryo-EM map for a RIOK2 containing particle that was done, was published by Katrine Kabstein and colleagues. And what we did was we took this pre-40S particles. They also had pre-40S particles in the absence of RIO2P. We generated difference maps, and we docked our model into the electron density using molecular dynamics flexible fit. And we were able to actually figure out the orientation of the RIO kinase on the pre-40S particles. And we see that the body, the kinase domain portion of the RIO kinase, or RIOK2, was bound primarily to the head domain of the pre-40S particle. And the winged helix domain, which is here, is sort of nestled in between, kind of near the junction between the two. And years later, a couple of years ago, the Roland Beckman's group published the structure of the yeast pre-40S particle with RIO2. And this is the structure right here. Our structure looks very similar to that. So we were able to tell back then that, what the orientation was and get some ideas about how RIO2 might work in the context of the pre-40S particle. So if you look at how RIO2 docks, a few things were striking. One is that the kinase, the active site, was oriented towards the RNA. So basically the RNA, and there was no protein nearby. So there was no protein that could be potential phosphorylation substrates. And there was, no access to the active site when the protein is bound. Furthermore, there's this loop, which we had known from looking at sequences is a highly basic loop, and it points towards the RNA. So we quickly realized that this loop is probably playing a role in interaction with RNA. And furthermore, there's a conserved lysine at the tip of this P loop that is present in all RIO kinases, and that was oriented towards the RNA. So just looking at that, we had some ideas about how to go about trying to figure out how this protein functions. So it turns out that, the other thing I forgot to mention that was striking was that the entire kinase domain was interacting with the RNA. So if you look at the sort of distribution of positive charge on the surface that would be facing the RNA, it's even more striking. You can see really this protein has lots of positive charge, really ready to interact with the nucleic acid along all of its domains, which was something that we didn't know going into this. We didn't realize that the RIO kinase surface is primarily used for binding to RNA. And this is just comparing, several different structures that we've solved of both RIO2 and RIO1. So from that structure, we generated a model where we propose that the RIO2 binds in the early maturation in the cytoplasm. You get this, as I mentioned earlier, this release of particles. But we knew that RIO1 perhaps would have to bind in the same location as RIO2. It was already known that if you pull down RIO2 containing particles, you don't see RIO1. If you pull down RIO1, you don't see RIO2. And that would mean that there's probably some sort of structural rearrangement that would prevent RIO2 from binding. And that made sense because RIO1 does not have the winged helix domain, while RIO2 does. And we also knew at that time that the mature pre-40S particle, in the mature pre-40S particle, there's a rearrangement of the head domain and the body that sort of closes up the space between them. So we proposed that the RIO1 would probably bind at the same site, but with a more closed pre-40S particle. And last year, it was published, again, Roland Beckman's group published a series of structures. This RIO2 containing pre-40S particle and RIO1 containing pre-40S particles that shows just that RIO2 binds for the early stage maturation. And then when it's releasing all of these factors, RIO1 binds in the same spot, just in a slightly, with a slightly shifted head domain. There's also this is a really great paper. There's information about how the helix that I mentioned, C-terminal helix, actually interacts on the RIO2, for the RIO2 structure onto the pre-40S particle. And so is no longer bound between the head, so the N-terminal lobe and the C-terminal lobe, as we saw in the free protein. So you can imagine that its purpose may be to prevent the turnover of ATP until you actually need it, which is when it actually docks to the pre-40S particle. We also learned some things that, for example, if you look at the docked RIO2 versus the free RIO2, there's an opening. Again, perhaps helped by the movement of this alpha helix that would normally be located between the two domains. And we looked very closely at different nucleotide binding states to try and figure out if there is a conformational change that is nucleotide dependent that we could really point to as, you know, the kind of gate between states. And so here's an example where we looked at the ADP phospho-aspartate complex and the ADP vanadate, which is in the cyan color here. And if you look at the two structures, they're practically identical. So moving to sort of this transition state mimic complex, we didn't see any real change that we could link to a change in state. We, at that time, didn't know that this helix was moved in an RNA-bound structure, but hypothesized that perhaps it could be playing a role because that helix actually inserts an arginine into the active site that interacts with the catalytic aspartate. And so, you know, we actually call this the inhibitory helix because it could play a role in both shielding the phosphate from hydrolysis and disrupting or blocking catalytic activity. That's just kind of giving you an idea of where it is again. So, and then that helix actually, I'm just kind of highlighting, it moves to this position. So it's very far away. And, you know, the linker is disordered from the RIO2 when it's bound to the pre-40S. So we also were interested in the catalytic cycles and how things might change as you go from an ATP bound state to an ADP bound, ADP phosphoaspartate state to the pre-40S. And so we were trying to understand these steps, and I didn't have time to put in all of the data, but basically what we found was that you get rapid conversion from ATP to this phosphoaspartate ADP step. What's slow is the release of ADP. So we proposed that this phospho-ADP structure of phospho-RIO2 ADP structure binds to the pre-40S, and there's a conformational change required to release the ADP from the pre-40S. And then that altered pre-40S particle can now bind to the RIO1 phosphoaspartate ADP complex, and then go through the cycle again to result in the final maturation. So we do see evidence for alternate states. So this is a kinase which is very atypical. So, you know, a lot of the things that, you know, those of you who are familiar with kinases know about the activation loop and the phosphorylation resulting in the organizing of the active site and creating an active kinase. A lot of those things don't necessarily work in this case because you don't have an activation loop, you don't have a phosphorylation step that results in activation. So we were trying to understand some of that. Very early on, we had a structure of RIO1 bound to adenosine and then one bound to ATP. And what we saw is that here, this is the metal binding loop, and this is a metal binding aspartate. And we saw in the presence of adenosine, you have a very altered loop because you have a peptide flip that orients this carbonyl in a different position, and that results in a difference in the conformation of that loop. And so we at the time, we didn't know why this was the activated state. But I think the answer is not, clear. But what we knew is that the ATP state was less likely to form oligomers than the adenosine bound state, and that we did that through a series of analytical centrifugation experiments. And so one thing we did know was that in the presence of ATP, you actually had a state that was more sequestered. So more recently, in our work in studying this ERG inhibitor, this is an inhibitor that was actually found to inhibit the growth of prostate cancer cells. We saw the structure of that. And if you compare these structures to other structures in the database and a structure that we generated, we began to see some clues about how this might work in the context of the human RIO kinase proteins. So this was work done by Nishad Siraj, who recently graduated. And this is the human RIOK2 with ERG inhibitor. This is not the first inhibitor RIOK2 structure. There's at least one other one in the database. But here's our inhibitor in the active site. And at first glance, it looks very similar to all of the other RIO kinases. The one thing I should say is that this flexible loop region that is disordered in every other structure we've had, for the first time, we were able to see the full structure of that loop. Now, we also had a structure of the RIOK2 protein bound to ADP and vanadate, again, trying to do this mimic of this potential transition state. And what we see there is we also see a movement of the M loop, or a change in conformation. But we also see a change in the P loop. If you look at this RIOK2 with bound inhibitor, it clearly forms a dimer that is facilitated by or stabilized by interactions of the flexible loop, which is why we were able to see that. But also kind of there's interactions near the catalytic loop and the P loop. So when we looked at our structure of ATP and vanadate, or ADP and vanadate, we did not have dimers in our crystals at all. These dimers were not found in the packing. And it turns out that the P loop is in a very altered position, and the flexible loop is disordered. And if you kind of look at things more closely, again, you see that this change in the orientation of the metal binding loop, the interaction between the metal binding loop and the P loop facilitates the formation of this dimer, placing in a conformation where it's able to dimerize. So again, you have this dimerization that is dependent on basically having a inhibited or an inactive structure. if you look at this protein, you also see something curious in how the dimer, what the surface distribution of residues are on the dimer. So here's just a electrostatic surface of the dimer, and basically now it's sort of flipped compared to every structure that I've shown you so far, where the winged helix domain is on the bottom and the kinase domain is on the top. And what you see is that it's very negatively charged on the top and very positively charged on the bottom. And that reminded me of transcription factors, where you sort of have this distribution of charge such that if the molecule is approaching the DNA, it would naturally flip towards orienting the positively charged residues towards a highly negatively charged DNA. So if you rotate this molecule upwards and then rotate again, you see these two, and this is sort of the this would be the axis of rotation for the dimer, twofold axis. What you see is that the distance between these two residues, which are highly conserved in the RIO2 kinases, is about 34.4 angstroms, which is basically the length of almost the length of a complete turn of the DNA. So this started a discussion with a collaborator who works on RIO2 as well, but looking at interactions in the, basically he was doing chromatin immunoprecipitation experiments. And he asked me, does RIO2 bind to DNA? And so at the time I said, well, I don't know. I don't think so. I suppose it's possible it does have a winged helix domain. But basically, once we saw this, we started thinking about those experiments. So these experiments are still underway, where they're trying to figure out the exact locus of interaction on DNA. But clearly there's interaction in chromatin precipitation assays. If you do a screen for potential binding sites, you don't really get very good hits in the experiment that have been done so far. But this is turning out to be an interesting story, just again, very much structure guided in what it is, what the hypotheses are, and what to look for in these experiments. So I'm going to basically take about 4 minutes to talk about what we've done for our anti-cancer strategies around targeting RIOK1. And I mentioned, previously all of the processes involved in ribosome biogenesis, which is highly upregulated in cancer cells. And there's a number of different molecules that are known anti-cancer drugs that have been shown to be active against various steps of ribosome biogenesis. And RIOK1 and RIOK2 themselves have been shown to be at the center of numerous tumor promoting pathways, such as AKT activation and RAS signaling, and PRMT5, which is now emerging as a target for anti-cancer drug design. So there's definitely, a lot of interest in blocking RIOK1 and 2. We know that if you block these, that results in a ribosomal stress response, which basically results in activation of P53. and if you look at this in the context of no P53, you actually see some P53 independent pathways leading to this ribosomal stress response. So it's an interesting target to go after, but there's also some intricacies that need to be worked out. So RIO1 also interacts with PRMT5, as I mentioned, and there's actually a lot of interest in the interaction site. This is just a model based on a PRMT5 structure docked with RIO-K1, but RIO-K1 is known to recruit nucleolin, which is important in ribosome biogenesis, to PRMT5 for methylation on arginines. So again, a long time, when I was a postdoc, I actually found some molecules that could potentially inhibit RIO-K1. This Toyocamycin and tangibamycin, they were already known to play a role in affecting rRNA processing. And so we tested them in the lab. We saw that based in thermal shift assays, Toyocamycin can actually stabilize both human and archaeal RIO1 by 12 degrees. We saw the structure with the archaeal protein and was able to generate some hypotheses on how to go forward with drug design. We also found that Toyocamycin stabilized the ATP bound form of the protein, so sort of active state. So my student Min-Kyung Kim, who also graduated recently, developed a synthetic scheme where we could use click chemistry to change, basically add on a number of different monomers to come up with a library that we could test against RIO1. And I can't really tell you about all of the compounds because it's still in the IP process, but we were able to generate a series of compounds that had the effect of preventing colorectal cancer cell growth in a cancer-specific fashion, and also inhibiting the PRMT5 RIO-K1 interaction, kind of suggesting that the ability of RIO-K1 to bind to ATP or to catalyze ATP hydrolysis was necessary for the interaction between RIO-K1 and PRMT5. So you can see here, we have a control, and in link 2 is basically the the PRMT5 protein that is pulled down by interaction with RIO1. If you look at compound 3, you don't see any disruption of this complex, but compound 4, you see disruption of the complex. And this is a transfection control. So I'm going to end here. I gave a lot of different things, and I'm open to any questions you may have, but I just wanted to kind of give you an overview of all of the different structure-based studies that we've done trying to understand the RIO kinases. And I really would like to thank a great group of students that I've had over the years, and my collaborators, Sebastian Ferrer-Silka and Ed Hood, as well as Aditi Banerjee. I didn't talk about all her work on the colorectal cancer cells, but she's done, amazing work looking at RIO kinase inhibitors, and Puru Puranik, who actually helped my student with synthetic chemistry because I'm not a synthetic chemist, and really a lot of collaboration on the RIO2 project. And so I'll take any questions. Thank you for your attention.
Thank you very much, Nicole. That was great. So You may either place a question in the chat or simply, excuse me, unmute and ask your question. I don't see any hands up at this point. Why don't you stop sharing your screen, Nicole, and then we can
Yes,
see
I'm
the
gonna do that.
entire audience in the grid view.
Okay.
So I was intrigued. So I'll exercise the chairman's prerogative and ask a question. So I was intrigued to see the presence of a winged helix domain fused essentially to the kinase domain.
Yeah.
And were you able to get a clear enough view of the interaction with the RNA to confirm that the mode of binding was similar to that seen for other nucleic acids?